Hello and welcome. I'm your host, Neil Howard. Thank you so much for joining us for another segment of Health Professional Radio. We'll be speaking with Dr. Fahar Merchant this morning. He's joining us here as President and CEO of Medicenna Therapeutics. Medicenna Therapeutics is a clinical stage immunotherapy company. He's going to talk about the company's Superkines platform. Welcome to Health Professional Radio, Dr. Merchant. Thank you for joining.
Thank you very much, Neil. Really happy to be part of the conversation today.
Tell us a bit about yourself and what led you to your current role at Medicenna.
Well, Medicenna is my fifth biotechnology company. It's, for me, I've been a serial entrepreneur from the past 30 years or so. The last three companies have been very, very focused in the oncology space. And oncology itself is close to my heart. It has been part of my family. To see a family member survive cancer is an amazing experience. And I think this is something that really keeps me motivated, keeps me excited to pursue developing new therapies for oncology.
As a biotech clinical stage immunotherapy company, give us an overview of your company's approach to developing these immunotherapy based on novel, highly selective versions of IL-2, 4, and 13 tunable cytokines.
Yes, so these interleukins, IL-2, 4, and 13 that you just mentioned are interleukins that are just generally present. Each one of us has these circulating in our bloodstream. They modulate or control our immune system. But these cytokines in some patients may not be present in the large amounts or not adequately to fight off pathogens or to fight off cancer, for instance. And at the same time, these naturally occurring interleukin 2, 4, and 13s may not be the right therapeutic agents. So the approach that we have taken is essentially modify these very, very slightly, just, engineer them in such a way that these naturally occurring interleukins become super interleukins or superkines, and essentially make them, transition them from not being therapeutic agents, but to become therapeutic agents that are more efficacious, that are more safer, and therefore allow us to modulate and manage the immune system in the most efficient manner so we can then use our own immune system to fight cancer and other diseases even more effectively. So that is the approach we are taking with the Superkines and focusing on these three that we've in-licensed from Stanford University.
Now, it's also my understanding that you're in clinical development in melanoma and some other solid tumor conditions, MNDA11. I understand that's your lead pipeline asset. Is that correct?
It is one of our lead pipeline assets. MDNA11 specifically is a interleukin molecule that has been, as I said, engineered so that it is very specific to target cancer-fighting immune cells. And at the same time has been engineered so that it is a lot more safer. Now, just to give a bit of background, interleukin-2 has been in the marketplace for about 20 years or so to treat patients with melanoma and kidney cancer. But unfortunately, the naturally occurring version of this interleukin-2 is incredibly toxic. Patients get this drug only if they are admitted into an intensive care unit because the side effects can be life-threatening. And second of all, the drug doesn't stay in the bloodstream long enough. So what happens is that patients have to be infused with the drug every 8 hours for 5 days in a row. So that makes it a big challenge to keep patients under surveillance in an intensive care unit for that longer period. So MDNA11 essentially has an albumin molecule attached to it, together with our IL-2 superkine, which is safer, much more effective, but at the same time stays in the bloodstream so much longer so that the patients get administered the drug once every 2 weeks instead of 3 times a day. And the other thing is also that by creating this particular molecule, MDNA11, and having the albumin, it sort of allows the drug to localize in the tumor, to localize in the tumor-draining lymph nodes. This is where you train and you recruit your cancer-fighting immune cells. So it's very targeted, but at the same time, very specific and a lot more safer than naturally occurring IL-2. So that's the program that's in the clinic right now. And we have seen promising data so far. We have not seen the sort of toxic effects that you would normally see with Proleukin or the naturally occurring version of IL-2. The other thing we've seen, although the purpose of a phase 1 clinical trial is really to demonstrate safety of the drug, but what we have seen surprisingly is that in some patients, 2 in particular, one with melanoma and more recently another patient with pancreatic cancer, to have tumor shrinkage being observed where all other therapies had failed. So really exciting data that we've seen already in the phase 1 setting. And here we are sort of getting ready for the phase 2 portion of the clinical trial. And hopefully what we've seen in the pancreatic cancer patient can then be translated to other patients as well using the right dose and the sort of a healthier patient population.
Explain to our listeners what an immunologically cold tumor is and how this therapy can be, I guess, extended to those types of tumors.
Yes, so immunologically cold tumor, I think a good example is the one that I just mentioned, pancreatic cancer.
Okay.
And pancreatic cancers generally tend to be tumors where most of the immune cells are excluded from the tumor. And in doing so, our immune system is essentially not able to notice that the tumor even exists there. So these tumors tend to hide behind some kind of a shield. We call them the tumor microenvironment, where the tumor is unseen by the immune system. By using a drug like our drug MDNA11, we are able to overwhelm the system by sort of creating a much higher population of these cancer-fighting immune cells, so that eventually these immune cells are able to penetrate the tumor, and therefore, once they start attacking the tumor sort of, which was originally cold, becomes inflamed, and that's when you sort of call that tumor has become hot. And this is where the immune system is able to notice, oh yes, here is a tumor, and therefore the immune system can go after it. So that's what the intent here is with a drug such as MDNA11.
Could you also talk about another candidate in your pipeline, MDNA55, that's being studied in patients with a very fatal form of brain cancer?
Yes, so this particular drug, MDNA55, is, as you mentioned, has been used in patients and completed phase 2 clinical trials, getting ready for a pivotal phase 3 trial. This particular drug is for patients with what we call recurrent glioblastoma. This is by far the most aggressive, uniformly fatal form of brain cancer. And we've tested this drug in patients where the survival expectations for these patients is between 6 and 9 months in the sort of the best case scenario for these patients. In our phase 2 clinical trial, what we were able to demonstrate is that with just a single administration or a single treatment with our drug MDNA55, we were able to more than double the survival outcomes for these patients from something like 6 or 7 months to over 15 months. So this is really exciting. Another program that we look forward to advancing with a partner. So the intent is for us to work with a potential commercial partner that has the wherewithal to market and sell the drug. And the final stage for the drug is really conducting this phase 3 clinical trial in this very difficult patient population that we've seen very promising results so far.
Well, Doctor, is there anything that you'd like to add for our listeners?
Yes, I think first and foremost, we have a number of key milestones coming in the next 2 to 3 quarters, and these relate to the clinical data from the phase 2 portion of our MDNA11 clinical trial. And these will be data from our single-agent treatment of patients with 3 different tumor types. We'll have those data coming out over the next 2 to 3 quarters. plus the data set where we are combining MDNA11 with Merck's drug Keytruda. So that's, I would say, that's the first set of bits of information. And the other area that I'd like to cover is that Medicenna, being based out of Toronto at the moment, we are really putting together a strong team at the sort of the biggest biotech hub in the world, and this is in Boston. And we've gone out there and recruited some key C-level executives that we will continue announcing, in addition to the chief financial officer, the chief business officer, and soon to be announced the chief medical officer. So looking forward to those key features of our company's expansion and growth as we advance a later stage into the clinic and build a solid C-level team in Boston.
Well, if you would, give us a website where our listeners can learn more about Medicenna.
Yes, the website is www.medicenna.com.
Dr. Merchant, I appreciate your time this morning.
Thank you very much, and a pleasure to speak to you today.
You as well. You've been listening to Health Professional Radio. I'm your host, Neil Howard, with Dr. Fahar Merchant. Audio copies of this program are available at healthprofessionalradio.com.au, also at Anchor, Spotify, and be sure and subscribe to our YouTube channel at youtube.com/healthprofessionalradio.